Hello students, welcome to Aakash. So today we are going to discuss the AIATS plus phase 2 paper mock test 3, which was held on 2nd of Feb 2025. Is that it? Like we are going to solve the question monotonously? No, that's how AIATS plus is different. So over here we are not only going to talk about the questions, that how to approach them, but also the entire periphery with which this particular question belongs, right? Because certain times when you read the topic, you got confident, okay, now I know this entire topic and now I can solve any sort of questions regarding it. But then a question comes that tricks you. And that's what we are going to deal over here, so that you will know the entire little nuances of that particular topic as well, right? So in this first session, we are going to talk about certain chapters and the questions that are being associated with it, and also going to talk about that how you're going to solve them, right? So the topics we are going to discuss today, the chapters are the principles of inheritance and variation, cell cycle, and also ecosystem, respiration in plants, and microbes in human welfare, right? So now let's start with the questions. So the very first question over here says prophase is marked by, right? So we have identified, okay, fine, this is a question from cell cycle, right? Now, when we talk about this particular prophase, from where does it belong, right? So if we talk about cell cycle, we could divide it into two phases, right? The interphase, and it's been popularly known as the resting phase, right? Because any the events that are being occurring during this particular phase cannot be observed under microscope, right? That's why we are going to call it as the resting phase. Someone is already answering, it's option 1. That's wonderful. We'll discuss it and then we let you know that whether you are correct or not, right? So till that particular time, so when we talk about this cell cycle, the another phase is going to be the M phase, where actual cell division takes place, right? So when we talk about this interphase, we could further divide it into G1, S, and G2 phase, while this M phase could be divided into karyokinesis, where the nuclear division takes place, and then cytokinesis, where cytoplasmic division takes place, and ending of this particular event will finish one cell cycle, right? So where does this prophase actually stands in this entire flowchart? So it falls over here in karyokinesis, where the nuclear division is being there, right? So if we talk about the characteristic event of prophase, right, then the characteristic events over here are the formation of mitotic apparatus, right? Then condensation of chromatin material, and another key event is going to be disintegration of nuclear membrane and nucleolus as well. And because this particular membrane is basically disintegrating, that is the reason why the endomembrane system also disassembles here. So that's been disappeared in this very phase, right? So you could clearly see over here that the centrosomes are now moving towards the pole. So certain time we got confused about the centriole, or we could say centrosome moving towards pole and chromatid moving towards pole, right? So these are two different phases. And apart from that, you could clearly see that over here, the condensation of chromatin material is also taking place, right? So if we move back to the question, then the question is saying that it is marked by. So we have to identify the characteristic event of that. So the very first one is saying initiation of condensation of chromosomal material. So that is the characteristic event of prophase, right? Then it says that formation of loose chromatin material. So when we say that it is the formation of loose chromatin material, that means rather than condensation, we talk about decondensation. And this particular event is a characteristic feature of telophase, right? So here during prophase, untangling occurs, right? This is a line that's been specifically mentioned in NCERT, that the chromosomal material untangles so that it could condense further properly. But on the contrary, over here in telophase, it gets detangled again, getting back into the chromatin phase, right, which is used to be there in the interphase kind of a thing, right. Then it says alignment of the chromosome at the spindle equator, right. This is the characteristic event of metaphase, right. So till this particular phase, the condensation is entirely completed. The disintegration of the nuclear membrane has already been done, and that is the reason now the cell shift from prophase to metaphase, where all the chromosomes that are now being completely condensed are going to align themselves on the equatorial plate, right? And finally, it says separation of chromatids, right? This is a characteristic event of anaphase, right? So as the question over here demands about the characteristic event of prophase, which is correctly being depicted in option 1. So that's how you will be answering this particular question. Hope this is clear to everyone, right? All right. So moving ahead to the next one. The next one talks about interkinesis. Again, a question from cell cycle, but this time this M phase karyokinesis is not going to be mitosis as the earlier question. This time it is meiosis, from which this particular phase belongs, right? So interkinesis is basically a very short-lived phase and it marks the interval between two meiotic divisions, right? That means not two meiotic division, I would say meiosis 1 and meiosis 2. The duration between meiosis 1 and meiosis 2 is being covered by interkinesis. Right? I'm so sorry about it. So this is the interval between meiosis 1 and meiosis 2. So the question that is being depicted over here is a very easy question that talks about that how exactly, this interkinesis represents the phase between which phases of meiosis 1 and meiosis 2. But certain times in the previous year questions also, it's been asked that what's the characteristic event of interkinesis, or what is the event that is different from the phases of the interphase, right, like S phase, G1 phase or G2 phase. So when we talk about this interkinesis, over here what happens is, in the animal cell we'll see the duplication of centriole, right, as we could see in S phase. Over here we could see RNA and protein synthesis as well, because the cell has to prepare itself for meiosis 2, right? Meiosis 1 is being completed, a little bit of decondensation occurs, the mitotic apparatus is already dissolved, so this is the time where all the preparation that needs to be done for meiosis 2 is going to occur. The only thing with which it differs from the S phase, because this is a characteristic event of S phase of the animal cell, the only difference it has that over here DNA replication does not occur, right? That's how it is different. So certain times if the question demands you to talk about the similarity of S phase with interkinesis or the dissimilarity would be over there DNA replication occurs, over here it doesn't. Similarity would be centriole duplication is a common event in both these phases, right? I hope this is clear to everyone. So someone is asking whether the separation of chromatid occurs in metaphase 1 or metaphase 2. So when we talk about chromatid separation, it occurs during meiosis 2, and it's because the separation of homologous chromosome occurs during meiosis 1. And meiosis 2 almost resembles mitosis. We know that in mitotic anaphase, separation of chromatids occur. Same goes for meiosis 2, right? So that's how you have to read the questions, read the topics, by identifying the interrelationships between different topics, like how they are similar, how they are different, right? So these are the events that occur during interkinesis, and as I have specifically mentioned that it is an interval between meiosis 1 and meiosis 2, so we have to see the last phase of meiosis 1 and the first phase of meiosis 2. So the last phase of meiosis 1 is going to be telophase 1, right? So in these particular options, we could rule out all these three, because this is representing prophase 1, telophase 2, prophase 2. This is the only option that is depicting the last phase of meiosis 1, that is telophase 1. And the first phase of meiosis 2, that's going to be prophase 2. So the correct answer is going to be option number 4, right? So this is an easy question, but yes, certain times it comes into a difficult category or maybe the medium category by asking about the similarity of these phases or differences of them, or maybe the characteristic events that is being occurring during these phases, right? So do pay attention while you are reading these topics, that don't skip the interrelation between the topics, right? I hope this question is clear to everyone. All right, so now let's move ahead to the next one. So this particular question says, in which of the following processes carbon dioxide is not released, right? So this particular question belongs to respiration in plants, right? So we will take up each and every reaction one by one, and then we come to know that whether carbon dioxide is released during the process or not, right? So in basic, if we talk about respiration, so we know respiration is all about making the energy for the cell in the form of ATP, right? And in order to do so, a catabolism has to be performed, right? In which we are going to break the bonds that have been existing between the carbons, and then we are going to use that particular energy in the formation of ATP, right? So we know that the preferred substrate for respiration is glucose. So we are going to, break those bonds and releasing every carbon in the form of carbon dioxide, and then utilizing that energy to create the ATP, right? So we are going to take each and every reaction one by one. The very first one says oxidative decarboxylation of pyruvate. Well, the name itself suggests carboxylation, that means something related with carbon dioxide, and because it is writing D, that means it is being eliminated out, right? So what happens during oxidative recarboxylation of pyruvate, popularly known as the link reaction? Right? So during this reaction, the pyruvate molecule, which is being yielded by glycolysis process after the cleavage, glycolytic cleavage of glucose. So pyruvate is being obtained. Now an enzyme known as pyruvate dehydrogenase is being utilized to catalyze this particular reaction. So pyruvate is going to employ coenzyme A and a reduced coenzyme NAD+ as well. So these particular coenzymes are going to be utilized by this enzyme in order to convert this 3-carbon molecule into, and this is a metal ion that's also been used by this particular enzyme, to convert it into acetyl-CoA, which is a 2-carbon compound, right? So we know that the reactions are always going to be balanced. So if it's 3 carbon, there is 1 carbon still left, because 3 carbon, 2 carbons are being utilized for the formation of acetyl-CoA. Now what happens with that 1 carbon? That is going to be released as carbon dioxide, right? And of course, over here, this particular oxidized coenzyme gets reduced as well. So it's going to yield NADH plus H plus, right? So we can clearly see that during this particular reaction, carbon dioxide is released, right? And this is this particular reaction is being performed so that pyruvate can gain access inside the mitochondria, right? That's how further processes like Krebs cycle or electron transport system could work out on this for the complete degradation of the glucose molecule, right? So this is a reaction where carbon dioxide is going to be eliminated. But the question is saying that during which it is not released. And what we have seen in this particular one, it's being released. So we could rule out this option. Then it says alcoholic fermentation, right? So when we talk about fermentation, so we know over here, unlike the aerobic respiration, here the complete degradation of glucose is not going to take place, right? Over there, it's a complete degradation. Over here, there will be partial degradation, or you could say partial oxidation of glucose, right? So during fermentation, specifically if we talk about the alcoholic fermentation, what happens is that again this pyruvate which is being yielded by the glycolytic pathway, right, is acted upon by an enzyme known as pyruvate decarboxylase, right? The name of the enzyme itself is clearing the fact that it is going to eliminate, D means elimination of carboxylation, that means carbon dioxide elimination is going to occur. So when pyruvate decarboxylase act upon pyruvate molecule, it is going to yield acetaldehyde, right? Similarly, like the above reaction, this is again 3-carbon compound yielding a 2-carbon compound, and of course during the process that one carbon dioxide, the remaining one is going to be eliminated as carbon dioxide, right? Now this acetaldehyde will be worked upon by alcohol hydrogenase, right? And during this particular reaction, NADH plus H plus will be used, and the enzyme gets this particular coenzyme gets oxidized into NAD plus and yield ethanol, right? Now, why this particular reaction is taking place is because you have studied glycolysis already. So during glycolysis, what happens is these oxidized coenzymes are required so that we could generate NADH molecules, right? Now, again, those reduced coenzymes in case of aerobic respiration are going to reoxidize back again by electron transport system, right? But over here in fermentation, because the entire process, whether it is glycolysis or fermentation, is occurring in the cytoplasm of the cell, right? So over here there is no oxidation mechanism, so that these particular oxidized coenzymes could be regenerated again. So this is a way with which they are regenerating the NAD plus so that they could be utilized again into the glycolytic pathway, right? So again, either way, in this alcoholic fermentation, what we have seen is the release of a carbon dioxide molecule, right? So if we go back, we could clearly see that during alcoholic fermentation as well, carbon dioxide is being released, right? Now, the next one we have is lactic acid fermentation. So, if we see this again, now we will talk about the lactic acid fermentation. Again, as it is fermentation, right, so there won't be the complete breakdown of the glucose, right? Again, it will be a partial one. But this time again, the key product of glycolysis, that is pyruvate, which is a 3-carbon compound, is acted upon by lactate dehydrogenase. And here again, the reduced coenzyme is going to be utilized and it will reoxidize it, right? And at the same time going to yield lactic acid. Now, different from the other two reactions, over here this 3-carbon compound again yielding a 3-carbon compound, right? So no carbon is being left that could be eliminated as carbon dioxide. So that is the reason why during lactic acid fermentation there is no release of carbon dioxide, right? So as per the question, this particular reaction, the lactic acid fermentation, is the reaction in which carbon dioxide is not going to be released, right? Now the fourth one is saying Krebs cycle, right? So when we talk about the Krebs cycle, so over here what happens is that during this particular cycle, what we see is that when the isocitric acid is converted into alpha ketoglutaric acid, this particular conversion occurs. So this is a 6-carbon compound yielding a 5-carbon compound. So obviously carbon dioxide is going to be released. And further, when this alpha ketoglutaric acid molecule is going to be converted into succinyl-CoA, right? This is a 5-carbon compound and this is a 4-carbon compound. Again, 1 carbon is being released as carbon dioxide. So these 2 times we could clearly see the decarboxylation reaction that occurs during the Krebs cycle, right? So we could clearly see that in these cases as well, carbon dioxide is being released. So on the basis of that, we could eliminate this particular option as well, because during this particular cycle as well, we can see the release of carbon dioxide, right? So as per the question, because the question is asking about the reaction in which carbon dioxide is not going to be released, that is clearly depicted in option 3. I hope this particular one is clear to everyone. Okay. So now let's move ahead to another question which talks about a dihybrid cross. That means this belongs to principles of inheritance and variation. So this question says that in a dihybrid cross of a true breeding round and yellow seeded pea plant with wrinkled green seeded pea plant, what will be the percentage of recombinants in F2 generation, right? So when we talk about this particular cross, this is a very classical dihybrid cross, right? So when we talk about this particular cross, so we know that the parents are, it's been clearly depicted that they are true breeding, right? That means they are pure lines. That means they are going to be homozygous for this particular trait or character, we could say, right? So they are being true breeding for round and yellow color seed, right? So the genotype that they are going to exhibit would be capital R, capital R, capital Y, homozygous for both the traits. And on the contrary, the other parent is going to show the recessive, phenotype, again true breeding. So this is going to be small r, small y, right? Now when we talk about the gametes they are going to form, it will be capital RY, and over here it will be small ry, right? And the F1 we are going to yield from them would be capital R, capital Y, small y, having round and yellow seeds. This is the F1 generation. Now, in order to yield the F2 generation, we know that we have to perform selfing, right? The self cross, which simply means that you have to cross the particular individual with an individual that is having similar genotype, right? So, we are going to cross them with capital R, capital Y, small y, right? So, when you have this particular cross done, what we are going to yield, the classical ratio of 9 is to 3 is to 3 is to 1, right? So, over here what we are going to see is, right, at least one copy, one dominant copy of both the alleles, right, and the other ones could be variable, right, but we are going to see the dominant allele for both the characters in these 9 genotypes that we have. In this particular case, if we're going to see, then one character will be present in its recessive form, while the other one is going to have at least one dominant copy, right? In contrast to this, this one is going to have the dominant copy of the other allele and the recessive of the very first one. And this is going to be the pure line. That's going to be small r, small y, right? Now, the question is asking about the recombinants, right? If we see the parents here, right, parents are having at least one dominant copy, right? Over here, both dominant copies are present. Here, both the recessive copies are present in the parents. Here, at least one dominant copy is present, right? So, this is the parental feature, that either to have at least one copy of both the alleles dominant or having all the recessive ones present over there, right? So, if we have to designate here the parental combinations, so this is going to represent the parental combinations, because they are going to have at least one copy. They could have both dominant as well, but they are having at least one dominant copy for both the alleles, right? So, these are the parental combinations. Same goes here. It's implicitly represents this particular parent. So, this is also depicting the parental combination. Now, what is a recombinant? Recombinant means that is going to be different from the parents, right? So, these ones are basically depicting recombinants. And now, why they are depicting it? Because they are having only one dominant copy of one allele only, which is not being present in any of the parents that we have used in this entire cross, right? So, this is depicting the recombinants. So, out of the 16 possible combination, 6 of them are recombinants, right? So, in order to identify that what will be the percentage of recombinants, so the total number of recombinants we have got is 6 upon the total outcome, that is 16 different possible combinations multiplied by 100, right, gives you 37.5%, right? Correctly depicted over here in option number 3, right? So, that's why simply these things could be ruled out. Certain times the question could be asked in terms of that how many of the offsprings, or what will be the proportion or the percentage of the offsprings that are going to have at least one dominant copy of one allele or of both allele, right? So, you could simply go by calculating with this particular ratio, that how many of them are going to attain that particular trait, right? So, do pay attention while you are reading this particular cross, because this dihybrid cross is really important and the question could be asked in any frame, right? I hope this particular question is clear to everyone, right? All right. So, let's move ahead to the next question, and the next one we are having is the exotype of sex determination, right? So it talks about sex determination. So when we talk about sex determination, a very popular type that we study is on the basis of types of chromosomes that an organism possess, right? So based on this, we could have two types when the males are heterogametic and when the females are heterogametic. Right? So when we talk about the male heterogamety, then we could have either the type XX, XY, right, where XX represents the female and XY represents the male, right? So this is how they are heterogametic, because they are going to produce two different type of gametes here. Other type would be XXXO type, where the male lacks a sex chromosome, right? So going to again produce two type of gametes, but one of them lacks the sex chromosome, right? Also, the number of chromosomes that are being found in females would be different from that are found in males. Males will be having one less chromosome than the females, right? So this type of sex determination, if we see it in humans, and this we could see in grasshoppers. Right? So if the question asks that in which of the individuals we are going to see same number of chromosomes in males and females and different number of chromosomes in males and females, we could clearly see different number of chromosomes in grasshopper but same number in humans, right? Now when we talk about the female heterogamety, then we can have Z, W, ZZ types. Over here this represents the female, this represents the male. Right? So over here males are homogametic, so they are not going to decipher the sex of the individual. On the contrary, over here females are going to decide the sex of the child. But in male heterogamety, male gametes are going to decide this particular thing. So when we talk about this ZW, ZZ type of sex determination, this could be seen in birds, and another type would be ZOZZ, where ZO represents the female and this represents the male. This could be seen in butterflies, right? So here also the number of chromosomes that are being found in males and females are going to be different, but this time females are having lesser chromosome than males, right? Now apart from that, we can have haplodiploid sex determination, right? And this could be seen in honeybees, right? In this particular case, the males and females are having different number of chromosomes. Also, the ploidy is also going to be different. Like over here, females are diploid, having 32 chromosomes, while males are haploid, having 16 chromosomes only. So in the next generation, if we talk about, then they are going to produce the gametes with the help of meiosis, right? And males, on the contrary, are going to produce the gametes with the help of mitosis, because further the number cannot be reduced. And meiosis is a reductional division. So over here, the gametes are going to be produced by mitosis. If in case the fertilization occurs, then it will yield 32 chromosomes. That means diploid condition. So this is going to result in the production of females. On the contrary, if this particular egg develops without fertilization, and the process would be the parthenogenesis, right? So if it develops without fertilization parthenogenetically, then it's going to yield 16 chromosome, the haploid condition, and it's going to produce the males, right? So this is how, if the question demands that in which of the individuals you are going to find different number of chromosomes, we can clearly see a lot many examples like grasshopper, butterflies, even honeybees. Similar number of chromosomes, we could see birds, humans, this kind of sex determination could also be seen in the model organism, which is Drosophila, right? You must have studied it while, reading the experiment of T.H. Morgan, right? So this is how the sex determination methods are being found in organisms. So getting back after this to the question, it says that exotype of sex determination could be seen in, right? So as per the chart, we have seen it that it could be seen in grasshoppers, right? So over here, it's correctly depicted in option 3. Then we can see birds where, as we have seen, we could see CCZW types. In honeybee, it is haplodiploid sex determination method. And in case of Drosophila, it will be XX, XY type, right? So this is how you will be solving the questions. I hope this particular question is clear to everybody. In case you have any doubts, you could put in the chat box so that we could discuss it. Any other questions? All right. Moving ahead to the next question. And this question talks about which of the following is an example of polygenic inheritance, right? So when we talk about the polygenic inheritance, the name itself suggests poly means multiple, genic means gene. That means there are going to be multiple genes that are going to control the inheritance of a character, right? So when we talk about the polygenic inheritance, there are 3 or more genes that are being involved over here. Also, it is not only the genetic imprint, right, that is going to decipher the entire phenotype in the organism, but it also takes into account the environmental cues. Right? A popular example, let's suppose, could be, let's say, height of the individual, height in humans, right? We could see a range of individuals with different heights, and certain times the nutritional, qualities and stuff like that is also going to decipher that what's going to be the height of the humans. It's not only the genetic imprint that is a human is receiving, right? Similar way, the skin color in humans, correctly said by so many students over here, that it's option 3. Skin color in humans also, shows a gradient of colors in humans, right? And that is also going to take the environmental cues, like what is the temperature outside. You could clearly see the grading difference, the color difference in case of the people residing in Europe and then the people residing in the equatorial areas, right? So that particular gradient can be seen. One more important point when you study the polygenic inheritance is that over here, whatever combination is going to be present, whatever genotype is going to be present, the effect of each allele is additive, right? This is a very important point. So it will be a cumulative effect of all the alleles that are going to be found in the genotype. So if, let's suppose, we are considering skin color in humans and we are seeing that, let's suppose it's been controlled by, let's say, 3 genes. So if all the dominant alleles are going to be present, the color is going to be the darkest. Reason being, all the dominant alleles are going to have explicitly amplified effect cumulatively, and because of that the melanin production is going to be huge and the person is going to have the darkest color. On the contrary, if the alleles that are being present in the genotype are going to be the recessive ones, let's say like this, because all of them are recessive, right? So there again effect will be amplified, and it's going to create the lightest skin color that is possible, right? Yes, more intensity means more number of dominant alleles, right? So this is how you will see the entire gradient, and each allele is going to contribute in the effect, the final phenotype that is going to be exhibited by the individual, right? Now, on the contrary, if we talk about the sickle cell anemia, it's going to be controlled by a single gene, right? So, what happens during sickle cell anemia is that in this particular disorder, the beta globin chain is not going to be synthesized properly. There will be a functional imperfection into that, because at the 6th position, there will be a substitution, mutation that occurs, due to which the glutamic acid is going to be replaced by valine, right? So, and because now a polar acidic amino acid, right, the glutamic acid, is going to be replaced, so sorry. So, because over here, glutamic acid, which is a polar amino acid, is going to be replaced by valine, a non-polar one, right? That is the reason why the biconcave shape is going to turn into sickle shape. I'm sorry, my sickle shape is still deformed, but that's okay. I think hopefully it should not be in an individual. Over here on the board, it's okay. So this sickle shape kind of a thing would be achieved, right? And because of this, when the hemoglobin undergoes polymerization, this particular thing happens, the oxygen carrying capacity of this particular RBC is also going to be reduced because of the lesser surface area and the shape that it acquires, right? Right, correctly said, crescent shaped RBCs could be seen, right? So, sickle cell anemia, the important thing over here is that you don't have to just read the causes, but also the inheritance patterns. Reason being that these disorders are not only going to be asked in the form of that how they are being caused, what is the reason for it, but also in the form of pedigrees. So, on that particular time, you should know that how exactly you are going to see the inheritance pattern of them. So, over here, if we talk about sickle cell anemia, it is being inherited in an autosomal recessive fashion. Right? So, this is going to be inherited in autosomal recessive fashion. Then we talk about phenylketonuria, which is being popularly known as, yes, you are right, Madhushri, it's going to be third that we have discussed. Now we are discussing the other, disorders as well, because the questions could be asked on these as well, not only in the form of a factual data, but also in the form of analytical pattern, in the form of pedigree, right? So when we talk about phenylketonuria, it's an inborn error of metabolism, right, during which the enzyme that converts phenylalanine into tyrosine is not going to be present in the individual, due to which this conversion cannot take place. This is going to accumulate, convert itself into phenylpyruvic acid and other derivatives, and this is going to create mental retardations and other health implications in the individual, right? So, phenylketonuria is also exhibit the inheritance pattern in autosomal recessive fashion. That means the individuals that are going to have this particular disorder is going to show homozygous recessive genotype for this particular trait, right? Now, the last one we have is thalassemia, right? So, if we see the similarity between sickle cell anemia and thalassemia, both are blood disorders. In both of them, hemoglobin is going to get affected. Over here, non-functional, or we could say lesser functionality of the hemoglobin is impaired. Over here, the synthesis is being impaired entirely, right? There will be a reduced synthesis of hemoglobin, right? And now, what particular chain is synthesized, like in a lesser amount, would be the case where, thalassemia, that particular thing is going to decide which type of thalassemia is there. So, if it will be a reduced synthesis of alpha chains, we are going to call it as alpha thalassemia. If it's going to be reduced synthesis of beta chains, it's going to be beta thalassemia. And this particular disorder also, have the inheritance pattern in autosomal recessive manner, right? So, as per the question, because it's only asked about the polygenic inheritance, the trait that exhibits this, that could be seen as here skin color in humans. It's not a disorder. Rest all are disorders, right, which are being there in the individual because of the imperfection in certain machinery, biochemical or physiological machinery, like this one for the synthesis. During the synthesis, and substitution mutation is occurring. This is because there is a mutation because of which the enzyme is not being synthesized, and this is because the hemoglobin chain itself is not synthesizing, right? So there will be reduced synthesis. Again, the question could be asked in this form as well, that how exactly these two are different. Like similarity blood disorders are there, beta globin, synthesis is going to be impaired if we compare beta thalassemia with sickle cell anemia. The difference is over here, this is a qualitative difference. Either the mutation is present or not present, right? And if this particular thing is going to be there, then it is a qualitative disorder. That means how much reduced synthesis we could see in the organism, right? So someone is asking, albino is not disorder. See, it is, but skin color is showing a gradient of it, and all of them are not the disorders. Albino case is because melanin is being involved in other physiological reactions as well, which is going to maintain the, accurate, or you could say optimum survival condition in the organism. So because over here there will be reduced synthesis, extremely reduced synthesis of melanin. So that is why, yes, albino is not going to have a normal life, but on the basis of that, we cannot consider skin color in humans as a disorder, right? Because there is a gradient of them. We, as an individual, also see, exhibit certain skin color, and we are, like, thankfully not diseased at all, right? I hope I have cleared myself. Is there any doubt still? Is this question clear to everyone? All right. Now, based on this particular one, this is how you will be solving it. Now, let's move ahead to another question, which talks about which of the following is true for quiescent phase, right? So, as the name suggests, quiescent phase. So, this is basically depicting the G0 phase, right? That means it is going to be static in terms of certain functions of cell cycle. So this is going to be static in terms of cell division. So basically, what is the characteristic of this phase is that the cell is still metabolically active, right? The cell has to be metabolically active. The reason for that is that the cell has to maintain its living state. For that, metabolism is an essential process, right? We have studied it during studying the characteristic of living beings. Metabolism is important. So over here, the cell is going to remain metabolically active, but it's not gonna proliferate, right? Proliferation is not going to occur until it's being required, right? Until if there is certain injury is going to be there or certain thing. So one more thing that has been came as a PYQ as well, that it exit G1 phase to enter into G0 phase, right? This is a very important line, comes a lot many times, that in order to enter into the G0 phase, the cell exit from G1 and then enter into G0, and when again it's been called for the proliferation, it exits the G0 to get back again into the G1 phase, right? So this particular phase is a quiescent phase, right? So if we talk about the options over here, the very first one is saying that cell is metabolically active. As few students are saying, this is option 1. So yes, over here the cell is going to remain metabolically active so that it could perform the functions that it is being destined for, right? So option 2 says cytoplasmic growth occurs for cell division, right? This is not the case. Now why? Because in this particular case, in this particular phase, if the cell arrests itself, there will be no proliferation. So if the cell doesn't have to divide, why it needs to prepare for it, right? So that is why it's an incorrect option. Then the third one is saying that cell replicates its DNA again and inappropriate option, because the replication of DNA is essential. Why? Because when you divide the cell into two daughter cells, both the cells require the copy of that genetic material. And in order to do so, you should have two copies. And in order to create those two copies, replication is required. So again, this particular event is required for cell division. But as we are reiterating this particular fact that the cell is not proliferating here, so the cell is not going to replicate its DNA in this particular phase. Also, this is a characteristic event of S phase, right? This is a characteristic event of S phase. Now, the fourth one is saying nuclear division takes place. Again, it's incorrect. This occurs during M phase, right? So, yes, cell replicates during S phase. Correctly written. Wonderful, right? So, when we talk about the true statement for quiescent stage, it's going to be that cell is metabolically active, right? I hope this question is clear to everyone. All right. Now, let's move ahead to another one, which says we have to read the statements and we have to select the correct ones for Morgan experiment on linkage, right? So, if we see the Morgan experiment over here, so he's been credited because he deciphered this term linkage, right? Also performed this particular experiment in order to define what it is for, right? So someone is asking, is it for class 11 student? So it is an entire syllabus of NEET having 11th and 12th chapters comprised of. So there are certain topics of 11th and certain topics of 12th in this particular session, right? Because this paper has been designed so that you can have a keen and a rigorous practice of how you're going to appear yourself in NEET, how you're going to perform there. So that's why it is having the topics from both of it, right? All right. So when he took this particular cross, he has took two characters. One is the body color, right? And the dominant one over here is going to be the yellow, right? And the eye color, which is white, right? So when he crossed this, right, what he has found here is that the parental combinations that he have achieved here are 98.7% in the F2 generation, right? And the recombinants that we have got is only 1.3%, right? So what we could infer from here when he has performed the crosses, that because the parental combinations are too high, that means the genes that are responsible for these traits that are being responsible for these traits are too close to each other, right? Like for example, if we consider this cross, what we are going to see that these two genes Y and W, because they are too close to each other, then because of this closeness during the time of gamete formation, it is difficult to perform recombination between them, and that is why these genes are sought together, right? So usually if we consider, let's say this case, right? This particular case, and we make the gametes out of it, so we consider that the genes are going to segregate, right? So all of the alleles could have possible combinations with each other. That means these genes are like this, right? So this R could come with capital Y, this R could come with this small y, this R could come with small y, and also with the capital Y, right? But now in here, if we see, then the genes are placed so close to each other, like let's say over here Y. So we are going to understand it with this R and Y example. So these R and Y are so closely placed to each other that this R, like many times, like if let's suppose 100 events are occurring, so 90% of the time this R is going to assert itself with capital Y, and this R is going to assert itself with small y, right? This segregation is going to occur rarely, right? because the recombination is not possible here. They are so close to each other. So here in this case also, what happens is, because of this close placement, what we are seeing is that the gene assort together, and that is the reason why parental combinations are too high, 98.7%. And the recombinants are too low. But on the contrary, if the genes are far apart like this, then what happens is parental combination reduces now and the recombinants increased in comparison to this particular cross. So if we are seeing in a question or in a cross that the parental combinations are too high, we could simply infer that the genes are so closely placed to each other that they are going to assort together and the parental combinations are going to be higher. And if they are far apart, then the recombination chances are possible, right? So this is how you will be having this particular cross, performed by Morgan. So based on this, if we see it, so when the genes were grouped on the same chromosome, some genes were loosely linked and showed very low recombination. This would be an incorrect option because they are saying that the genes are loosely linked and still showing low recombination. That's not possible. Then it says that the genes for white eye color and yellow body color were tightly linked and showed 1.3% recombination. So yes, when we see the white eye and yellow body color, they were tightly linked. So this is a correct statement. Then it says the genes for white eye color and miniature wings were loosely linked and showed 37.2% parental types. That's incorrect. 37.2% were the recombinants. And then says Morgan found that proportion of parental gene combinations was much higher than non-parental gene combination. This is true for both the crosses. So this is how you will be selecting these statements. I hope this question is clear to everyone. So that's all for this particular session. Please stay tuned for the next one as well. Thank you very much.